It will cover everything that we've covered so far in the course, including today's slides. And it will be available all day on Wednesday. You'll have 75 minutes to complete it once you start. And it is a closed book. All tests in this course are closed book. So that means that there will be no regularly scheduled class on Wednesday. So don't try to sign in for Zoom on Wednesday at 12:30 because there won't be any. You'll be using that time sometime during the day to take the test. Okay, everybody clear on that?
So don't sign into Zoom on Wednesday, basically.
That's right. Don't sign into Zoom. Just take the test, and that time commitment is your time for this class on that day.
Okay, thanks. Just wanted to clarify. Thanks.
How many questions will be?
Number of questions. What's your name?
Agarid Al Yusuf.
Okay. I have to be sure to give me your name anytime you speak so that I can record you present. How many questions? There are 20, I think roughly 25 questions.
25, 17 minutes.
75 minutes.
75. Okay.
So it's a full class period.
Oh, sounds good. Thank you.
That would be scary. 17 minutes.
100 questions, 10
Thank
minutes.
Excuse me. I didn't know if you already said this, but can we take the test anytime during the day, or is it a specific time?
It's any time between midnight and midnight.
All right, perfect. Thank you.
That was Adnan?
Yeah, late Adnan.
Is it everything we've talked about so far? Is that what the test is
Yes,
about?
yes, including today.
Thank you.
I also have a question. This is Marissa. You haven't let out a study guide, is that correct? There is no study guide.
No, the worksheets are study guides.
Oh, okay.
Anything else?
I had a quick question about the worksheet due tonight. I think second to last section where we draw the structure of each molecule. Did you want that like in Lewis structure with the orbitals, or like just circles or something?
Something that it makes it clear to me that you know what the question is asking. So different students have submitted a lot of different formats, but most of them have been, you draw circles for orbitals and then you put dots on them for electrons. And some people try to draw all the neutrons and protons, but that's not necessary. If you just write a note on the side, like 25 protons, then I know that you know how many protons are in that nucleus.
Okay, thank you.
Yeah, don't forget to turn in your worksheet because it's worth a maximum of 35 points, which is not insignificant. Okay, let's see. The textbook pages for this section on the worksheet are 39 to 54, if you want to refer to that. And I sent you another assignment on biomolecules. It's a grid, little boxes for you to fill in. And that's due next Monday, March 1. Okay, I'd like to do a couple of things right now. Can you see quiz 2 on the screen? Okay.
Yes.
Yes.
Yeah, so we've had 2 quizzes so far, quiz 2 and quiz 3. Quiz 1 was not graded. And I'd like to go over the answers, make sure that you're straight on the answers to the questions on both these quizzes. First of all, because you should know them. And secondly, some of these questions are relevant to the test coming up. So pay attention. On quiz 2, the first question, match the best definition with the term. Okay, control is a constant in an experiment. Paleontology, fossilized life. Homeostasis, stable equilibrium. A molecule is chemically bonded atoms. And a theory is an explanation with general applicability. The next question was about publication. You've got some exciting results. You want to let the rest of the world know, and what is the least appropriate way for disseminating scientific information? Well, a number of items are listed there, but the least appropriate one would be to send a letter to the editor of a newspaper. And why is that?
Zach Newschuler here. For one, it's scale. It's not going to get out to very many people, so it'd be very difficult to get out to the scientific community at large. But also, it generally letters to the editor are opinion pieces and are not fact.
Good point. They're opinion pieces. Yeah. The main thing here is that all of the other organizations or scientific organizations or having to do with science. So essentially, if you published or presented a talk, it would be to peers who could knowledgeably review your work. Editor of the newspaper is a journalist, very unlikely knows much about science. So that would be the least appropriate type of person that you'd want reviewing your work. Okay, number 3, cell is a basic unit of atomic structure. That's false. What's wrong with it?
The smallest unit of atomic structure is an atom. So the cell would be the basic unit for life.
Right. The word that should have been there is life, not atomic structure. Okay. Scientist determines a DNA sequence, and what scientific discipline is going to be used to analyze that sequence? And the answer is bioinformatics. That is the scientific discipline that is really devoted to extracting information from DNA sequences. So even though some of you said molecular biology or information technology, those aren't incorrect, but bioinformatics is the specific subdiscipline. Which ones of these are characteristic of pseudoscience? Well, resists criticism, driven by ideology, looks for information that supports a conclusion that's already been derived, and starts with a conclusion, then looks for evidence to support it. Then the next one, you overhear someone saying, my theory is that vaccines will completely control the coronavirus. Is that a theory? And it definitely is not, because there are no data to support the statement. And just for example, we now know that many variants of the coronavirus are coming out, and for some, there probably will not even be a vaccine available at this point. Then number 7 in science, pronouncement reflecting new discovery. Yay, I already found it. That's false because it's more commonly no reaction, or I don't understand why that happened. Now, what are the properties of life that are characteristic also of fire? Well, what I had in mind here was energy processing and growth. Quite a number of you had those and included response to stimuli. And I gave you credit for that because after thinking about it for a while, I can see that you can construe fire as having a response to a stimulus like a gust of wind. Okay, so check your quiz and make sure you got credit if you put response to stimuli. Then there's the graph. The independent variable is time, the dependent is velocity. And finally, an experiment is a test in which there's at least one treatment and at least one control, and that is true. So any more questions or comments about quiz 2? Okay, then I'm going to take quiz 2 down. Stop sharing that. And now you should see quiz 3. Do you?
Yes.
Okay. So the first question, multiple answers, indicate the statements that are components of the cell theory. Well, they are. Cells can only arise from pre-existing cells. Cells are the basic units of life, and all living things are composed of cells. Well, some of you included cells evolve over time to create new cells, and that isn't wrong. It's not incorrect. Cells do evolve over time, but you don't need to evolve over time to be a cell. And that statement is not part of the cell theory. So you didn't get credit for that one. Okay, match the specimen with the type of microscope best suited to observe it. Okay, a plant cell would be a compound light microscope. Bacterial ribosomes, transmission electron microscope. Fruit fly antennae surface features would be a scanning EM. Internal structure of a virus, transmission EM. And a living amoeba would be a compound light microscope. And that is a significant distinction because with any kind of electron microscopy, you can't look at living specimens. You have to kill them and fix them in some way in order to look at them with an electron microscope. Whereas with a compound scope, you can look at living materials. Fill in the blanks. Genes in eukaryotic cells can be found in an organelle called the nucleus. In prokaryotic cells, genes are in the nucleoid. And then there was this question number 4 on binomials. And how do you write binomials? So the first time it's mentioned in a scientific manuscript, it's spelled out. This is a bacterium, Bacillus subtilis, and it can either be italicized or underlined. And if it's underlined, there's a space in the underline between the words. Subsequently, after the first mention in a paper, it would be abbreviated. Bacillus would be abbreviated to just B, but subtilis is spelled out. And again, it could be either italicized or underlined. So pay attention to that, because it's going to come up on Wednesday on your test in a different form, but you need to know how to write a binomial. True or false, fungi acquire food by secreting enzymes and absorbing the digestion products. That's true. Then how are things classified? Into the domains are archaea, eukarya, and bacteria. I think almost everybody got that right. And then about Goldilocks, the things included are an atmosphere, liquid matte core, acceptable size, and liquid water. Any questions about any of the questions on quiz 3? And it's important that you know the answers to these because much of this will come up again in a different form, either in the next test or subsequently in the semester. Okay. If nothing else, I'll go on. To the topic of the day, which is biomolecules. Everybody see that? Biomolecules? Somebody
Yes.
say? Okay. What are the biomolecules? Nucleic acids, proteins, carbohydrates, and lipids. 4 classes. And they can be broken down. They're all polymers. That means they're long chains of subunits. So nucleic acids have subunits called monomers, and they are nucleotides. Proteins have monomers called amino acids. Polysaccharides have monosaccharides, whereas lipids have fatty acids. So these are the monomers that build up the polymers that constitute the biological molecules that are found prominently in all living things. So we'll take a look at all of these in turn. First, the nucleic acids. There's DNA and there's RNA. DNA makes up the genome, the gene stores the genetic code, whereas RNA helps to construct the proteins using the information that it finds in DNA. So the 2 nucleic acids work together. So let's consider how DNA is constructed. Each of them is built from nucleotides made from just 3 main components. The first is a phosphate group. So if you look in the diagram, the yellow circle is a phosphate group, and it's repeated here 4 times. So it's PO4. The other component is a 5-carbon sugar. That's the blue. Sugar with 5 carbons. And in the case of DNA, that sugar is deoxyribose. In the case of RNA, it's oxyribose. So it position 2 here in DNA. This shows the DNA form is only a hydrogen there. If this were RNA, it would be an OH at that position. And the 4th component is a nitrogenous base. And there are 5 possible nitrogenous bases. And they're shown here in the gray on the right side of the diagram. So In DNA, you have thymine and adenine, cytosine, and guanine. And if you look carefully, you see that the structure of each of those is different. Whereas in the case of the sugar and the phosphate, the structure is the same. In every repeating unit, the structure is the same. And so a nucleotide is the three-component structure: the phosphate, the sugar, and the nitrogenous base. So remember that. That is what constitutes a nucleotide. And nucleotides just repeat endlessly in these polymers. Now, the other point to be made here is that in the case of DNA, the base is thymine. In the case of RNA, it is uracil. So DNA does not have uracil, it has thymine. RNA does not have thymine, it has uracil. Everything clear so far?
Yes.
Yes.
Okay, good. Now, DNA is a double helix. It's double-stranded, composed of two helical chains wrapped around a central axis. And the important thing here is that the nucleotides are complementary. The nitrogenous bases are complementary in the two strands. So A always bonds to T, and G always bonds to C. You can see that here on the left, that in every case of an A on one side, there's a T on the other. So there's a G on one side, there's a C on the other. And it's important to emphasize here that these A's and T's and G's and C's are joined by hydrogen bonds. Remember, we spent a lot of time on the importance of hydrogen bonds, and they are weak bonds, whereas the other bonds joining nucleotides are covalent. They're strong bonds. So joining a phosphate to a sugar, that's a covalent bond. Joining a base to the sugar is a covalent bond. But when you get to the next form of the structure, when the two strands of DNA come together, they're joined, they're held together by hydrogen bonds, which are weak. Compared to covalent bonds, hydrogen bonds are weak, and that's important because in the process of metabolism, these bonds have to break. And we'll get to that later in the course. Now, what are the differences between DNA and RNA? I've said some of them already. DNA is double-stranded, RNA is single-stranded. DNA contains thymine, RNA contains uracil. RNA molecules are short, whereas DNA tends to be long, has to be at least the length of a gene. And DNA has hydrogen bonds, but RNA does not. Now, why is that? Let's look at these pictures. Why doesn't RNA have hydrogen bonds?
Hi, my name is Taija. I think that RNA only has no hydrogen bonds because it's only a single strand, so there'd be no need for a connection.
Yeah, that's right. It has nothing to hydrogen bond to because it is only a single strand.
Yes, sir.
Okay, and the other bonds in both DNA and RNA are covalent, as I said earlier. At the top of this diagram, you can see pictorially the difference between RNA nucleotide and DNA. In the yellow, that's uracil, whereas the green in DNA is thymine. And RNA, as I said earlier, at this number 2 position, has a hydroxyl, whereas DNA has only a hydrogen. So those are the key differences between the two. move on to proteins. What is a protein? It's a polymer composed of amino acids. So if you look at the bottom of this picture, you can see a representation of a protein. It's a polypeptide. The amino acids are hooked together in a chain. And the colored ones, alanine, glutamine, and isoleucine, are shown here at the top. And they're joined by peptide bonds, which are made by what's called a dehydration reaction. A dehydration reaction means a loss of water, dehydrated. And where does the water come from? It comes from a hydroxyl on one amino acid and a hydrogen on the other. So if you take these two out, a hydrogen and a hydroxyl, and make water, what's left is a bond between the carbon of one amino acid and the adjacent nitrogen atom of the next amino acid. So that what you see down here is a bond here between the C and the N, and then there's another dehydration reaction causing another bond between the C and the N here. So that's the way amino acids are linked together to make proteins. And what is important is that you recognize that all amino acids are alike in the sense that they all have an amino group and they all have a carboxyl group joined to a central carbon. And also joined to that carbon is what's called an R group or a side chain. And that's what makes the difference between one amino acid and another. So you can see that here on the right of this diagram. The top is the amino group and the carboxyl group joined to this side group, which makes it a tyrosine molecule. Or if you have the same amino group and carboxyl group, but a different side group, it's now glutamine. So the only difference among amino acids is the side group. Now I want to emphasize you need to know what is a peptide bond and how it's formed. So just to emphasize the point, you have two amino acids sitting adjacent to each other. Facing one way is a carboxyl group on one amino acid and an amine group on the other amino acid. A water molecule is taken out, and what that leaves is this carboxyl, C double bond O, is here. This NH is here, and a bond has formed across here. That's this. This is the peptide bond. This bond between the C and the N, and it's a particular C, C double bond O, particular N, and H. That peptide bond repeated over and over and over again between amino acids makes a protein. Well, here they are. Proteins are composed of 20 amino acids, and each, as I said, is defined by its side chain. And what you'll notice is that among all the amino acids, they all have this same amine and carboxyl, and they differ only by what's in the white boxes, the side chains. And there are 20 of them. Just want to make one additional point here that we'll come back to later, and that is that two of the amino acids, methionine and cysteine, have sulfur in them. Now, sulfur has a special capability of forming bonds, and when we get to the folding of proteins have to fold in order to be active. So in order for them to fold properly, most of them need sulfur bonding. And they get that from the fact that methionine and cysteine, 2 amino acids, each has a sulfur atom in it. Okay, amino acids and human health. Of the 20 amino acids, our bodies can synthesize half of them, 10. The other 10 we have to get from the food we eat. And these are called essential amino acids. And they're listed here. I don't have to read the names. But these are essential amino acids that we need to get from our food. And so some proteins are called complete proteins. They contain all the essential amino acids. And that means most animal products, eggs, milk, meat, fish, poultry, any of these have all the amino acids. Other proteins are incomplete. They're missing some of the essential amino acids. And these are found in plants. So for example, rice, wheat, oats, and other plants have no lysine. Beans are missing methionine and tryptophan. Peas are missing methionine, and corn is missing lysine and tryptophan. However, if you're a vegan or a vegetarian, you need not despair, because all you have to do, if you had a diet of nothing but rice and beans, it would be complete. Why is that? Because although rice is missing lysine, beans has it. And although beans are missing methionine and tryptophan, rice has those. So if you ate just these two types of food, you'd have complete proteins. Okay, next we do carbohydrates. Carbohydrates are composed of just 3 types of atom, carbon, hydrogen, and oxygen, in a ratio of 1 to 2 to 1. So the general formula for a carbohydrate is CH2O. The names of sugars always end in OSE, OS. Glucose, sucrose, ribose are examples. Monosaccharides. The monomer units of carbohydrates are called monosaccharides, which means a single sugar. Glucose, for example, is a monosaccharide. These molecules are very soluble. They're good sources of energy. They're important building blocks. They're often part of other molecules. So for example, we've just talked about the fact that DNA and RNA both have monomer sugars in them. In the case of DNA, that's deoxyribose. Most sugars have more than one form. In the case of glucose, it can be either a linear molecule, which you see here, it's a 6-carbon linear molecule, or it can be cyclic, as you see here. In solution, these two forms are at equilibrium. So they're constantly switching back and forth between the two forms in solution. And the equilibrium is highly biased toward the cyclic form. 97% cyclic, 3% linear. And you need to know what is an isomer. Glucose, fructose, and galactose are isomers. That means that they have the same chemical formula, C6H12O6, but they have different structures. And what you see here is an illustration of that. This carboxyl can change positions from being terminal to being interior in the case of fructose. Now, this seems like a very small and a very subtle change, but it can have a big effect. So for example, in the case of glucose and fructose, fructose is sweeter than glucose. And that's why, if you look at labels on your sweetened food that you eat, you'll almost always find that it's been sweetened with fructose rather than glucose or some other sugar. That's because fructose has this carboxyl on an interior position and makes it taste sweeter. For example, high fructose corn syrup is very commonly found as an additive in food. Disaccharides are two monosaccharides linked together. Examples are maltose, lactose, and sucrose. So here you have the formation of maltose from two glucose molecules. So again, to repeat, there's a dehydration reaction. You have the one carbon of one glucose molecule and the number 4 carbon of an adjacent one. They undergo a dehydration reaction, lose a water molecule, so you end up with this structure. The number 4 carbon is hooked to, the number 1 carbon is hooked to the number 4 carbon through an oxygen atom. So that's how disaccharides are formed. And another product of the reaction, of course, is a water molecule. Now, polysaccharides are long chains of monomer sugars. And good examples are starch, glycogen, and cellulose. The interesting thing about these polymers is that they're all polymers of glucose. So what makes them different? Well, they differ by the linkages between the sugars, between the monomers, and by their branching patterns. So let's consider starch, glycogen, and cellulose here. Starch has long chains that are branched. There are also some unbranched chains. And the linkages between the monomer units are either alpha 1,4 or alpha 1,6. It can be either. And I'll tell you what those are in a minute. Glycogen, on the other hand, instead of long molecules, they're short and they're highly branched. But again, the linkages are just like they are in starch, alpha 1,4 and alpha 1,6. Cellulose, on the other hand, is quite different. It has long chains, not branched at all. And what holds cellulose together is hydrogen bonding between the adjacent chains. Now, the other difference is that the linkages in cellulose are beta 1,4 instead of alpha. So let's take a look at what that means. Sugar linkages, alpha versus beta, and 1,4 versus 1,6. So glucose can have an alpha configuration or a beta configuration, and it depends on where this hydroxyl on the number 1 carbon, which side of the molecule it's on. If it's on the side of this adjacent hydroxyl, it's called alpha. If it's on the opposite side of this hydroxyl, it's called beta. So that is what distinguishes an alpha linkage from a beta one. And then 1,4 versus 1,6. Well, 1,4, the carbon atoms are numbered starting on the right-hand side with 1, 2, 3, 4, 5, and 6. So 1,4 linkages are between the 1 carbon on one molecule and the 4 carbon on the adjacent one. Now, what makes it 1,6 is if there's a branch. So now you have the number 1 carbon of one chain linked to the number 6 carbon on the adjacent chain. So that is a 1,6 linkage. And again, either 1,4 or 1,6 can be either alpha or beta. So that's just a comment about the nomenclature that you'll see when you read about polysaccharides.
Excuse me, Professor, it's Terrence Belprez. I just had a quick question in regards to polysaccharides. I don't know if you know, but I know like when in relation to when you consume monosaccharides, such as just simple or disaccharides like table sugar versus the polysaccharides like starches in pasta or rice. One of the things interested is like, of course, glycemic index, like at the rate your body absorbs the or it breaks down into just simple sugars. And like I've heard different things about, starches such as like, oh, they spike your blood sugar just the same as like a pile of candy does. But at the same, I've heard other people say no, it's a slow releasing carb, so it doesn't spike it. I was just curious, didn't know if you knew whether like at the rates.
Yeah, it's a good question, and I'm not a nutritionist, so I don't really know much about this. But what I do think I know is that if you eat monosaccharides, it has a very direct route into your bloodstream. So if you eat a spoon of glucose or fructose, you should feel, you could feel more energetic very quickly. Whereas if you're eating a highly complex polysaccharide, like starch, glycogen, or cellulose, these things have to be, all these linkages have to be hydrolyzed enzymatically. And that takes a much longer time. So the glucose doesn't go directly from your digestive system into your bloodstream. It spends more time in your digestive system getting broken down into monomers. And
So I guess,
it is the monomers that end up in your bloodstream, but it takes longer to get there.
so that is to say, like maybe each starch, like rice or pasta, have like a different strength of bonds, rate of being broken down, than say like highly fibrous foods, with a lot of cellulose, such as brown rice or like whole grains.
Yeah, and you mentioned brown rice. There are also pigments in food, and pigments can interfere with the digestion. So that's why brown rice has the reputation of being slower to break down because it has a brown pigment that interferes with enzymatic action. Scott, do you have any insight on this issue?
As I just put into chat, basically the way it breaks down is if you have packaging with glycemic index number on it, the higher the number, the faster these things are metabolized and broken down and enter the bloodstream because they're more easily digested. And the lower the number, the longer it takes. So the slower you get that blood sugar rise. And it's essentially what you were saying. It's just there are some packagings include a glycemic index number, and that's what the number would mean. And I think the cutoff is around 60. Anything above 60 is considered high. Anything below 60 is generally considered low.
And I was Oh, sorry. And I was just wondering if like the glycemic index, like, whether it's high or low, whether it has to do with like the strength of the bonds of the polysaccharides. I was just wondering, like, if you say it has a high glycemic index, would the polysaccharides have like weaker bonds? Is what I guess was what I'm wondering. It's like that's how I interpret it.
Well, the bond strength is the bond strength. The covalent bonds, all of them have the same bond strength as long as the molecule is the same. So for instance, all of the alpha 1,6 bonds are the same strength as another alpha 1,6 bond. The alpha 1,4s, they're the same strength as all the other alpha 1,4s. A lot of it has to do with the branching, because the more highly branched a polysaccharide molecule is, the faster it can be broken down. If you think of it, they basically get broken down one molecule at a time, starting at a free end. So if I have a highly branched molecule with a bunch of free ends on it, I could theoretically have a piece of molecular machinery breaking down all of these things at the same time. Whereas if I have something like a starch that is a single strand, I can only start with one thing at one end and work on down the chain. So it tends to break down slower.
Okay, that makes sense. That makes perfect, better sense. Thank you, Scott.
You're welcome.
And that's why cellulose is so hard for us to digest, because it's not branched at all. The chains are linked by hydrogen bonds, and so you just have these long chains of glucose molecules. And as Scott said, that it usually starts at one end. So that makes cellulose difficult to digest. But Scott, another question I have, or maybe anybody might know the answer to this, is it more difficult to break an alpha 1,4 or an alpha 1,6, or beta 1,4 versus alpha 1,4? Are these different linkages more or less difficult to hydrolyze?
I'd have to do a search on that. I don't know off the top of my head.
Yeah, I'm sure it can be found somewhere.
I
would
could
think
hazard a
that-
guess,
I would-
but my guess is that the energies are very similar. Like we're probably talking like sub-joule amounts.
Yeah. I would guess the same thing, but I don't know. I would just.
The other thing I would tend to believe is that a lot of whether it's broken down, the bond strength is probably similar, but it would require different enzymes to be able to match with the substrates, match the shapes of the substrates to get in there and actually do the breaking.
Mhm.
So probably the rate would be dependent on how much of particular enzyme you have, which is another reason we can't break down cellulose, is we don't have the enzymes for it.
Okay. Anything else about polysaccharides or how you derive nutrition from them?
So real quick, just to sort of distill this down a little bit, alpha 1,4 linkages are straight chain, alpha 1,6 linkages are branched.
Yeah.
Specifically, the alpha 1,6 is the branch. That is where it branches off. It's if you think of it like a node.
Yeah. And so here you can see that 1,6 is at the branch point, whereas 1,4 linkages are within a chain.
And you can also see on the top that anything past the branch point has gone back to an alpha 1,6. So you've got a branch and then another straight chain.
Alpha 1,4 here.
Yes, 1,4. Anything past the 1,6 is now an alpha 1,4 again.
Okay, let's move on. This is just a pictorial showing the branching. As Scott was saying, you can visualize now if an enzyme comes along and is nibbling at the ends, it has a lot of ends in a highly branched molecule, whereas in cellulose, each chain is unbranched. And so an enzyme has to start at at the end of each chain independently. And now our final topic on biomolecules is lipids. Contain mostly carbon and hydrogen. They're hydrocarbons. Linked by nonpolar, covalent bonds. So they're stable, very stable molecules because of the strength of their bonding. They're defined by their solubility properties. They're mostly nonpolar, which means they don't mix well with water. Water molecules are polar, so they're hydrophobic, tend to be greasy or oily materials. Functions of lipids, well, they're good at storing energy. For example, fat. That's where we store energy in our bodies, mostly. They're essential components of membranes, and we'll talk a lot more about membranes later. They can be waterproof coverings around cells, or they can provide insulation and cushioning of organs and tissues. So lipids have very important biological functions. Types of lipids, they're fatty acids and triglycerides. Triglycerides are the fats and the oils. The phospholipids, steroids, and waxes. So there are 5 different types of lipid that you should make note of. So they're acidic fats. So as I said, they're hydrocarbons primarily. So you have a long chain of carbons, fully saturated with hydrogens, and at one end, there's a carboxyl, and this is what makes it an acid. This carboxyl moiety at one end of the hydrocarbon chain. These are rigid molecules, hydrocarbons, if they're saturated. And saturated means no double bonds. And this is an example of palmitic acid. And this is rigid, and whether it's rigid or not becomes important in membrane formation. Now, if there is a double bond in the chain, it's now called monounsaturated with one double bond. And this is a flexible molecule. At the point of the double bond, it can bend. In this case, it's oleic acid. Again, an acid at one end. If there are 2 double bonds or more, it's called polyunsaturated, more than one double bond, as in linoleic acid. So then that brings us to what are fats. Fats are triglycerides. So what happens is that a molecule of glycerol, which is a 3-carbon molecule with 3 hydroxyls, with a hydroxyl on each of the carbons, again can undergo a dehydration reaction with the carboxyl end of a hydrocarbon, and that brings the hydrocarbon, it bonds to the glycerol molecule. And if that happens on all 3 of the carbons in glycerol, you have a triglyceride. So in this case, you have 2 saturated hydrocarbons and 1 unsaturated, which bends, and that takes up a lot more space due to the bending. And again, we'll come back to the importance of space later on. So this would be an example of a fat molecule. Next, the phospholipids. Phospholipids are composed of, they have three components: a nonpolar tail and a polar head composed of a phosphate group and another compound, another group containing nitrogen, which has a positive charge. The phosphate group has a negative charge. And of course, the hydrophobic tail has no charge. And the hydrophobic tail can be either saturated or unsaturated. So this, keep in mind, is the structure of a phospholipid, which is critical to the development of life to begin with and its maintenance. So phospholipids are peculiar in that they have these opposite ends. So if you make a mixture of water and oil, the oil will float and there'll be a sharp margin between the water and the oil. Now, if you pour into such a mixture phospholipids, what will they do? They will orient themselves so that their tails, their hydrophobic tails, are in the oil and their polar heads are in the water. Now, this is the basis for a membrane. So if you imagine in the prebiotic ocean, you have these molecules floating around, and over time, they learn to stick their tails in oily substances and their heads in water. Water is always present. So membranes formed in this manner. Without membranes, there could never be cells. Now here you can see that. On the left is a picture of a membrane. And if you look at its environment, membranes are in aqueous environments. There's water on either side of the membrane, but within the membrane itself, it's completely dry, completely hydrophobic. So the two tails come together and make this hydrophobic environment. The heads are pointing out away from the membrane. If you imagine that this is a membrane on a cell, the outside of the cell is aqueous, water. The inside of a cell is water. And the membrane separates the interior of a cell from the exterior. So that's why they're critical for life. You can't have a cell without a membrane. Another form of lipid is a steroid. Steroids are characterized by this 4-ring unit. It always looks like this. It can have a lot of modifications to form various different kinds of steroids. For example, here's testosterone, here's estrogen, and here is cholesterol. You see that they're distinguished by their substituent groups. Lipids that act as hormones are called steroid hormones. A hormone is a substance that is produced in one location and acts in another location. And that is true of the sex hormones as well as other types. So it's the side chains that make individual steroids distinguished from each other. And finally, the last type of lipid is a wax. It's one fatty acid chain linked to an alcohol. That's a wax. It's more hydrophobic even than fats are, completely insoluble in water. And biologically, it provides waterproof coverings. So for example, leaves depend on waxes for a number of their functions, as do fruits and feathers and of course, in our ear canals, we have wax. Now, this is an example of a wax structure and how it's formed between an alcohol and an acid. These both alcohol and acid are long polymers. But again, you have a dehydration reaction between the two, and they're joined by a covalent bond through an ester group, and you end up with this long molecule, which is very hydrophobic. Okay, that's an introduction to biomolecules. Anyone have questions or comments about this? The slide set is in Canvas. You can access it, and I will make the recording of this session available as soon as it's processed.
Excuse me, Professor.
Uh-huh. Yeah.
What page is in the textbook again?
Pages 39 to 54.
Thank you.
I think I said earlier that the biomolecules would be covered on the test on Wednesday. If I did say that, it's not true. This lecture will not be covered on Wednesday. So don't spend your time studying this until the next time. Anything else? Scott, you have a workshop coming up.
Yes, I do.
So if anybody wants to get a different perspective on any of the topics to be covered on the test, go see Scott and his workshop.
So if I may throw something in here, I noticed I get email tagged when people try and join the workshop and I have not started it yet. I start at 2. So I think Wednesday I had 5 people or so try and join before 2, and then I never saw anybody at 2. I don't start the meeting up until 2 or maybe a minute before, but please, if you didn't come in or if you couldn't make it in because you were early, I will open it up unless something comes up, in which case I would try and tell you in class. So please try and come back at 2 o'clock or after 2. Don't think that just because you couldn't join at first that the meeting wasn't being held or the workshop wasn't being held. That was not the case.
Okay. Anything else? Remember, there'll be no Zoom session on Wednesday. You have all day to take your test. So I will see you again next Monday.
Will the chemistry be in the test as well?
Yes, chemistry will be in the test, but not biomolecules.
Okay, thank you.
The worksheet, I'm looking at it and I'm not sure if we covered all this stuff. So all of this stuff will be in the book though, correct?
Yeah, I think so. Yeah.
Okay, because we definitely did not go over the gene that causes cystic fibrosis.
No, not yet. That's coming. No, that slipped in there. Are you looking at the biomolecule grid?
Yeah.
16 boxes?
Yeah, that's the one that I'm looking at that's due next Monday.
I'm going to have to change that. I thought I had taken the cystic fibrosis question out. And if I didn't, I need to put in
There's a couple on genes in there, so.
Yeah, okay, that's the wrong one. I have to change that out. Thank you for finding that. I'll change it as soon as class is over.
So it should be updated by what, like 4 o'clock?
Yeah.
All right.
before 4. Okay, anything else? Okay, well, then I'll see you all on Monday, next Monday.
And if I don't see you, good luck on your exams.
Yeah,
Thank you.
everybody aim for 100.
Have a good weekend.
Okay, see you.